Okay, well, let's get started. So I'd like to introduce Regan Baird, who is the Global Research Program Director at Visiopharm. And he'll be talking about turning images into knowledge from histopathology to multimodal spatial biology. And so we thank you for attending and presenting your work. Thanks.
Thanks, Evan, and thanks to the University of Michigan for giving me this opportunity to speak to you today. I'm very excited to be here and share with you some of the things that we are doing at Visiopharm. It's a pretty exciting time in spatial biology. And I just wanted to start with a little bit about who and what we are, and then jump into some of the recent publications that have been put out there using our technology across the spectrum of spatial biology. And we'll do it that way, I guess. And at Visiopharm, we really are embracing the artificial intelligence technologies in tissue analysis and tissue image analysis. And really the core of what we're going to talk about today is in dealing with tissue slides, whole tissue data. And we understand that there's advantages in applying AI in this realm in terms of being able to provide faster results, more robustness, reproducibility, but more importantly, to achieve the sensitivity, specificity, and precision that is necessary to get data out of tissue, data that you can trust, data that you can rely on, and creating workflows that incorporate artificial intelligence as well as your human intelligence working in concert together to get the most out of the data that you're generating. We see the application of AI in digital pathology in 3 realms. The one that I'm going to focus on today is in the preclinical research arena, which we color blue. The holy grail of digital pathology is for us to be able to provide tools to the clinics in human diagnostics to help pathologists more quickly uncover things and really push the boundaries of what can be done in diagnostic pathology. And a new area that we're pioneering is in stain consistency and stain quality, and using AI to make sure that those staining devices are giving you consistent results on a day-to-day basis, because after all, it is that staining that is going to be giving us all of the interpretations of the data that we're getting upstream and downstream in the process. Thank you for the lights. So when we think of spatial biology, we're all enamored by the latest indoctrination of spatial biology being these high-plex, multiplex images, thousands of transcripts, tens of thousands of genes. But really, there's a large realm of technologies that are at play when it comes to spatial biology. And for me, it always started with the H&E. H&E is the original spatial biology. It's really a morphometric approach. This is what our pathologists are using. That they go to school for a long time to really understand what's going on in these tissues. And they're doing it through visual interpretation of the morphological relationships of all of the objects in that tissue. With recent advances in technology, we can then look at many more biomarkers on those tissue through proteomics. There's a bunch of different imaging techniques that are used in proteomics. I've listed a couple here. I probably won't be comprehensive in my lists because there's always a new technology coming out on a daily basis. But as we're going from the morphological look at the tissue, we can also go into the proteomics approach. We also want to know what genes are being expressed in every single cell. So we're going to start looking at things at the transcriptomic level. And we're starting to see a bunch of technologies come out today that are incorporating tissue images with the transcript information so that we're able to create these bigger pictures of understanding of what's going on at the transcript level, but also at the proteomic level within the morphological look at the contextual relationship in the tissue. And I guess the genomics has actually been there for quite a while in being able to understand why some genes are being expressed, not expressed. Is it due to mutations? Is it due to some other things? It's probably the least amount of imaging happening in the pure DNA genomic side of things, but we're starting to see a lot of technologies start to flourish there as well. So let's start with histopathology. And so we've already been able to realize this potential of AI-driven precision pathology. In the clinic, we have developed what we call zero-click apps that can solve really challenging problems in the clinic. A lot of what we've developed is really around the breast cancer and the breast panel, where the pathologist will sit in front of the screen and everything is already analyzed for them already. We've developed really robust AI that can detect micrometastases in lymph nodes, looking at some complex biomarkers like PD-L1. We've developed algorithms using the same toolbox that our researchers use to create really, really robust algorithms. And the reason why we say they're zero click is because, for example, if we look at Ki-67 in a proliferation assay, we don't want to just find every positive nuclei in a piece of tissue. We want to find only the nuclei in the relevant part of the tissue, in the active tumor, which means we need to run an AI that not only finds the nuclei, but a step before that, that's going to find the tumor and discriminate between normal epithelium, which pathologists do really well, and lesion. And we only want to analyze the lesion. So we've been able to define an AI that could go in and do that separation of the different sections of the tissue automatically. And we also know in the clinic that on a piece of on a slide, there may be patient tissue, but also control tissue. So we need an AI to be able to distinguish between the patient tissue and the control tissue. So we create these analytical pipelines that can be run immediately. So when a pathologist or a researcher sits in front of the computer, all of the annotation is done already for them, including the results of the analysis. And these can be used in those patient decisions. Those can be used and put into your data tables or whatever you need to do. They're also modifiable if you disagree with what the AI generates. And we've got a couple examples of these. I won't mention the name of the university where this work was done, but it is a Big Ten school not too far away from here, where they were able to take our metastatic app and use it in an indication that it wasn't designed for. And what they found was that it had perfect sensitivity and a net negative predictive value. There's a word missing. A net negative predictive value in being able to really have confidence that the patients that you said were negative didn't have any of these micrometastases. And the one thing that keeps a lot of pathologists, breast pathologists, up at night is they said that a patient was negative, but they actually had some small ITCs or infiltrating tumors, cells that they missed. And so this really gives them the safety net to be able to apply an AI just to make sure on those negative patients that the AI isn't picking up anything. And they can work together to say, oh, that's real, that's something to worry about, or that's not real, that's not something to worry about. They've also taken advantage of our HER2 algorithm in breast and being able to show a increased read time and using our HER2. They've also found that using the AI, they get results similar to what's done in FISH and in reflex testing. So really being able to apply some AI to expedite the rate at which patients are getting the information that they need in order to understand where their diagnostics are going to be. These are really clinical examples, but these algorithms are also relevant in what we're doing preclinically in research. And so we can also apply these, or this kind of an idea where we want to apply an analytical pipeline that uses AI to solve our problems. Now, you might not have a problem with metastatic circulating tumor cells. You might not have a problem with HER2. And so what we've done is we've taken some of those algorithms from the pipeline or created some additional algorithms, and we create what we call template apps. And these are algorithms that you can use and you can apply generically for what you're trying to do, depending on the different areas. And you can string them together to create your own analytical pipeline. So for example, you may want to first have some type of large scale tissue detection so that you can isolate the areas that you want to analyze. Then you may throw in an algorithm that goes and compartmentalizes that tissue into the relevant areas before you do your cellular analysis. And once you've done your cellular analysis, you may want to go in and do some other quantifications based on the analytics that you get there. And you can string together the sequence that you would like this to work so you can batch process that across the larger data set. These are also all modifiable. It may not work perfectly for your data set, but you can simply add some more information or make some changes to what it's found in order to be able to perfect it to your research and your science. Here's an example where we were working with Dr. Berman in Queen's University. I think that's a safe one to mention, right? Up in Canada. And basically, we were able to run an algorithm that was able to identify every cell in an H&E. Some of these cells are mitotic. And so what we will do is we will just tell it, okay, these are the mitotic cells, learn that those are the mitotic cells, and it's a simple click to change that. And once you've learned, then let's apply it and make sure that you're able to find those mitotic cells on itself and on examples that it hasn't seen, and we iteratively give it more and more information so that it's able to find the different types of objects that we want it to find. Incidentally, we also had it learn what the difference was in bladder cancer. The David Berman story is really exciting for me because this is a pathologist who, looking down the barrel of his microscope, noticed differences in the different staging of bladder cancer patients. He just couldn't quantify it. And he said, I need an AI that'll help me understand the differences between these patients. I know physically I can see this, but fundamentally, I don't want to pull a ruler out under my microscope and start measuring every single cell. So we were able to give him the ability to find and quantify all of these cells. And then using a variety of metrics, he was able to hone it down onto which metrics of the nuclear morphologies were relevant for staging. And he's trying to redefine the staging paradigm for bladder cancer. This is done in TMAs, and now we're starting to apply this into whole cells, a whole tissue, and make sure that it works in the whole tissue arena as well. So there's more to come from that story. Now, there may be cases where we don't have a template app that solves what you're trying to do, that doesn't fit the needs of what you're trying to do. And with that, we've created a pathway so that you can actually start from first principles, and you can feed it annotation data, and it will learn from that annotation data the rules that you're giving it in order to create this automation. So all you need to do is provide it your expert knowledge. You feed it annotations. Those annotations you may have already, to be honest, and it will learn from the information that you're feeding it what to do. And so what we can do is we can take a sample that we've never seen before. We can feed it annotation information so that it can find every single one of these muscle fibers. It will then go in and annotate all of the muscle fibers for us based on the rules that we've given it. And then we can apply a second algorithm to go and quantify all of the nuclei, whether they're central or peripheral nuclei in these muscle tissues, using the algorithms that have already been defined. And so it'll go in and now using a predefined algorithm there. So we can modify algorithms to fit our needs. We can create new artificial intelligence to fit needs that don't already exist. So it's a very exciting platform to be able to work with because you're interacting with these images on a regular basis. And I know I've shown you a lot of things in chromogenic space, in bright field space. We're going to switch into fluorescence, which probably you're more comfortable with in a single cell world. But everything I've shown you here works identically as it would in the fluorescence space. So as we move out of the histopathology part of this talk and into the proteomics area of this talk, I want to introduce you to Amy Heimberger at Northwestern University. She's studying glioblastoma. They just published this paper recently. I think it came out actually while they were in, while she was at MD Anderson. But looking at the proteomics and the protein expression is important, but we also want to understand the spatial relationship of all of the cells that we're looking at. And so she's identified through an 8-plex the ability to find a variety of different biomarkers related to specific cell types. And what was interesting was that they started noticing co-localization of 2 of those different cell types at a regular frequency. You may notice some of the names on this paper, by the way. Some University of Michigan people are on this paper as well. And then looking to see if there were cell-cell interactions that were happening at a regular frequency. They even were able to find some triads or 3 cells all interacting at a regular frequency. And what was really important was understanding the distribution of these interactions in relation to the whole tissue. So it's not just are these interacting, but are they interacting in the tumor? Are they interacting nearby the tumor? Are they interacting far away from the tumor? And then also, are we getting larger and larger clusters of these? So understanding the spatial relationship of many, many cells requires obviously more and more signals, more and more biomarkers. So we're starting to see regularly in the field now things like this where we're getting common 8-plex immuno-oncology panels being built. And so can we start to define AI to help us understand and elucidate what's going on in these kinds of things, not only looking just for the staining, but also the cells that make up that staining, the cells that are comprised from that, and who is upregulated, who is downregulated, and where are they contextually in relationship to the XY location. And I love the fact that down at the bottom here, I don't have a pointer, I'm sorry, it says macrophage, like there's only one kind of macrophage, right? And we were working with Heather Stevenson Lerner at UTMB, where we actually appreciate that there are many, many different types of macrophages and many different types of macrophage markers. And so she's looking in liver, and she's really interested in understanding the difference between fibrosis in liver, cirrhosis in liver, or hepatitis C in liver. And just by looking at the macrophages, they can start to create diagnostic-like tests to understand the difference between the staging of these types of patients. But with an 8-plex, she only can just look at the macrophage markers. So we really want to start to be able to populate more and more biomarkers into the analysis that we're doing so that we can get a true picture of what's happening in the tissue. It becomes ever more complicated to start to analyze these kinds of data, because every biomarker you add in a proteomics study gives you an exponential increase in the number of combinations that you can have. Sure, that's not foreign to this group, but we decided to then rethink how digital pathology and spatial biology are going to play with each other through a product that we call Phenoplex. And our Phenoplex product is designed to meet the needs of that multiplex image analysis space. We had 4 mandates when we developed Phenoplex. By the way, I think it's close to the 1-year birthday of Phenoplex, which we're pretty excited about. So it's in its 2nd revision, and it's a very powerful tool for doing proteomics and spatial biology. But the first was to allow you to work in a single platform, a singular platform that has all the tools necessary to be able to get the data you need out of these very, very complex images. But it was based on the foundation that Visiopharm is known for, which is our ability to identify objects in tissue. So being able to do those regional compartmentalization of the tissue, but also being able to find every single cell. We need to know what you are, and we need to know where you are in the tissue before we can do any true spatial biology. Once we have that information, then we can go in and really gain some important information at the single cell level. We also said that it has to be able to handle any type of proteomics multiplex. Doesn't matter how many biomarkers you have. It doesn't matter how many images you're throwing at it. It doesn't matter how much, what the file sizes are. It has to work in all plex levels. You can't be limited by the number of things you're looking at. And it has to work in a stable environment, of course. And so really that was an important mandate for us. And then finally, for me, the most important thing is you have to be able to trust the results that you're getting out of this environment. You're not just going to trust the AI. You're not going to just trust the results. You need to have a way of interpreting and working back and forth with the data that you're getting out of these complex images, which leads me to this brief video. Hopefully it plays well.
Imagine being able to go from a library of multiplexed images to verifiable single-cell spatial data with just 4 buttons. The comprehensive Phenoplex workflow allows you to visualize your multiplex data, classify your important tissue regions, provide superior cell segmentation, expertly phenotype each cell, verify this single-cell data, explore the cellularity of the tissue compartments, uncover differences between cohorts, relay those discoveries back into the images, create the visuals for publication, and trust your spatial biology to Phenoplex, a complete workflow for all your multiplex image analysis needs.
Phenoplex has really been designed to bridge that gap between the bioinformatics and the imaging. There's this space in the middle where the biologists need to be able to visually interact with the images and understand what's going on in the image, but also interact with data tables and plots and a lot of other types of things. And so what we wanted Phenoplex really to be was an immersive, interactive experience where you can get your hands and see what's going on in that data as easily and as quickly as possible. It works with a variety of different instruments. This is only a subset of the instruments that we are able to work with, but pretty much any of the major instruments that are out on the market, Phenoplex works with. And the workflow is as follows. First of all, the first half of the workflow is about detecting objects in tissue, which we're really, really good at Visiopharm. We have very strong and robust algorithms that do this. The algorithms that will provide you, if they don't work, are all modifiable, so you can make them work for yourselves if you need to. I always recommend just trying it to see how it works before you go into that realm of trying to modify them. But in order to modify them, you just need to provide a little bit extra training set or maybe change some of the rules that it's following. The second half of the workflow is really about understanding the data and working and playing within the data, using some of the plotting tools that are available, scatter plots, t-SNE plots, different types of things, which will interactively allow you to really take a deep dive and explore within these complex multiplex images. One of the first things we did with Phenoplex in the early days was working with David Rim at Yale. This was 2 cohorts, one which responded to Herceptin treatment, so breast cancer patients that responded to Herceptin treatment, and patients that had a recurrence of breast cancer after Herceptin treatment. So basically, these patients all were deemed as suitable for Herceptin treatment. However, many of the patients didn't respond to treatment. Why was that? So they took samples from all of these patients, created a tissue microarray, and applied one of the very first image mass cytometry panels to those to see what was going on across 18 different biomarkers. They gave us this data. We simply ran our cell segmentation. We asked it to plot t-SNE plots. If you don't know what a t-SNE plot is, it's basically taking those 18 biomarkers, looking at their expression profile in every single cell and squashing it down into a single plot so that we can look at all of it simultaneously. And by plotting the responders versus the recurrent population, we could quickly identify areas that were similar between those biomarkers in the 2 populations and areas that were different. And what was exciting was within a few minutes, it actually took me longer to make this PowerPoint slide than it did to generate the data for these graphs, believe it or not. The HER2 receptor was obviously deficient in the recurrent patients. This is the receptor that the drug is going to attach itself to. And because it didn't attach itself to the receptor, we had a deficiency in the downstream signaling through beta-catenin, which was actually a novel discovery that they hadn't seen before, hadn't realized. Now, these were IHC positive for HER2, and we're learning today that there's different HER2 populations and HER2 low and different categorizations of patients that are happening in this HER2 space. So it's pretty exciting. But the real concept here is, okay, now how do we treat these patients that aren't reacting to Herceptin? Or how do we find a drug that both populations will respond to? Or more importantly, how do we get those cancer cells to trigger a beta-catenin response? Once we've been able to identify a plot like this, really what we want to do is we want to know where those cells are that have been gated in those circles, right? We want to see them. Right now, they're just points in a graph. I want to see them. And the interactive plotting tools within Phenoplex really gives you the ability to dive into these images. And so you can gate on objects in the t-SNE plot. You can separate the plots into different cohorts. Maybe you're interested in the cancer tumor regions, or maybe you're interested in glomeruli or whatever other compartments you have. You can facet these plots into different categories. And when you gate on objects in the plots, it'll show you a gallery of every single cell that you've gated on. So it's going to take a snapshot of that object and show it to you. So you can actually look at the biomarker expression profile of those cells. Is it expressing in the nucleus where it's supposed to? Is it expressing in the cytoplasm where it's supposed to? Or do I have a really bad segmentation and I need to go in and alter my segmentation? It'll also show you where those objects are in the tissue. So back on the image, it'll put a box around every single cell that you've gated. So it'll show you the spatial relationship of all of those objects and where they are in the tissue, all in a live feed, all happening simultaneously. What's very exciting about this is that if you have more than one image, because I don't want to analyze one image at a time, it will pool all of the data from all of the cells that you've analyzed, so that as I'm gating on cells, it could come from various images. And in a multi-view, I'm able to see the relationship of all of those cells across multiple images, not just one image at a time. We've also done something when you're setting phenotype positivity thresholds, that we create this co-expression table, which allows you to see from your control biomarker populations, what is the number of objects that are co-expressing two of your biomarkers. And if they should co-express, that number should be high. If they aren't high, obviously there might be something wrong and you need to go back and take a look. If they should be non-proportionally related, that number should be low. And if that number is high, then you may say, oh, do I have something going on with my staining? Do I have something happening with my segmentation? And being able to find all of those cells by clicking on a point in the data table and being able to find all of those objects in the tissue. So it's this interactivity and this immersive experience that we want to give you when you're playing with your proteomics data. Now I want to switch gears and start talking about transcriptomics, because this is actually where the field's starting to get pretty exciting. Well, we're very excited about what we can do in the proteomics space, but how can we link that to what's going on the transcriptomic side? We can see the proteins that are being expressed, but what's happening at the genetic level on the transcription? What genes are actually being expressed in those cells? And while this is hard to do in images directly because of the resolution of microscope images, we do have some content that can do this for RNAscope and for FISH and ISH and those fun things, if that's what you're at. But really, what's exciting is what we're going to be announcing at AACR in a couple of weeks in San Diego, and that's our relationship with NanoString, with their digital spatial profiling device, the Geomics. You can make parallels with MERFISH or 10X or anything if you want, because all of that technology is going to be overlapping for what we're talking about. We just happen to do it with NanoString and their Geomics platform to begin. And really, it's how do we incorporate artificial intelligence in the digital spatial profiling side of things. I don't know how many of you are familiar with the Geomics device, but the data that I'm going to share with you comes from a poster that we did with Kelly Hunter at ProPath and his team, where they were able to incorporate the use of AI to actually inform the device on where it should be doing things and to provide it more sophisticated segmentation tools. The way these devices work is really you're going to have to create what's known as areas of illumination. These are the areas where you're going to do your transcript analysis, and you have to tell it where those areas are. In the Geomics device, that's done simply by thresholding on one of the 4 colors they give you. In this case, we're thresholding on an area that's half tumor and an area that's half non-tumor. And then you select from those areas specific regions. So the regions are going to contain the different areas of illumination. That's going to release all of the transcript information, and you can read that on a sequencing device. Now, what we can do when we bring in AI is a couple of things. First of all, we can help on that segmentation side because artificial intelligence uses more than just the intensity thresholds. There's a common problem in a lot of the genomics instruments in that you have a high autofluorescence. Red cells have a high autofluorescence and the cells of interest may not be as bright. So it's hard to threshold those out. But with AI, we can simply eliminate some of those things because we're going to look at this, all of the signals of that object. We're going to look at it morphologically, and we can provide some help in doing AI-assisted segmentation. And there's a whole host of things that this opens up that we'll share with you in a minute. It's also available to be able to help identify where those regions are best suited. The example in the bottom middle here is looking at hypoxic areas of a tissue, which is really hard to see by visually. But if we can identify hypoxic areas of the tissue, then we can help identify areas where running the genomics instrument might be best suited. This is also useful if you're doing things like making TMAs and you want to know how to punch TMAs. This might be a useful technology there. Ultimately, when you run your genomics experiment, you've done this type of illumination and you've gathered data from areas, but you don't know how many cells of each type contributed to that area. And so using image analysis after acquisition, we can then go in and tell you how many cells there were, what types of cells there were. So you could standardize the results coming off of that genomics as well. So you can have more standardized region to region correlations. We've also wanted to introduce the ability of using H&E in the genomics space. So it creates a beautiful fluorescence image, but a lot of pathology and everything happens in H&E. A lot of that morphological investigation happens in that H&E space, and it can be pretty hard. Oh, I don't want you to go that far ahead of me, guy. Come back. So being able to use selection criteria from a variety of different sources, not just the image that the genomics creates. And here's an example where using AI, we could identify hotspots of tumor and then select areas that were half tumor and half non-tumor, or some combination of those. And then using co-registration technology, bring that together so that the AI is now suggesting what areas we should do our geomixing in. And I don't want that to go again. So this co-registration technology is something I'm going to share with you a little bit time. Our co-registration is an elastic technique. It doesn't require the nuclei to overlap. It's using serial sections. If you have serial images or serial stained images, it will also work there because you do have that overlap. Sorry, keep going backwards. And there's a lot of other really cool applications that we can talk about offline when it comes to being able to co-register information from different imaging modalities. And once that information is fed into the genomics device, then, and the AOIs are fed in because we have better segmentation tools, we really have a nice bi-directional integration going on with the genomics device, where we can feed it information. I don't know why the animation keeps running. We can feed it information to inform it on what it should be doing. We can use this co-registration technology really to enhance the experience if we're working with pathologists or people who prefer to work in the H&E space, or we want to put the data back onto an H&E, for example, we can do that. We can use image analysis to standardize by the number of cells that are being analyzed in a particular area or region. But we can also use all of that data backwards and bring that back into our platform so we can train more AI. We can use that information to improve the artificial intelligence because you have really nice annotated data already. And this co-registration technology is now starting to go berserk. I don't know if that's the right word. Working with Jared Burks at MD Anderson, he came to us a while ago and said, hey, I have this standard established immunofluorescent panel that we use on our Akoya system. And we don't want to redefine it for any other imaging, but we need more biomarkers. And so we've got this imaging mass cytometer device. Can we just use our standard immune panel and then co-register stuff coming off of the IMC and create a virtual 31-plex? So unfortunately, we said yes, and we had to do that. We wound up doing that with them. It was quite successful. And what they were able to do was find specific cell types using their immune panel and look at the functional markers that might be surrounding it using the IMC panel. Now, we didn't get perfect one-to-one co-registration because they were serial sections. But what was really cool about this was the fact that the immunofluorescence is coming in at a resolution and a size that's different than the imaging mass cytometer, even a different orientation. And the tool was what allowed us to do a fairly decent co-registration where the results were believable. We are now working more closely with Standard Bio tools to make this seamless, like we have been with the nanostring and the geomics. So that work was done with Sue. And then Sammy came along, and Sammy said, "Hold my beer. I'm going to take this to the next level." I guess she said, "Hold my wine." And she said, "All right, I want to start to incorporate more information. So I want to do not just imaging techniques from the proteomic side, but I also want to bring in a timsTOF image. And I want to do some metabolomics, which is a very low resolution technology, but it gives you many, 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 many more channels." And then they also brought in some transcriptomic stuff with the new Stomix device. And this paper was published last week. So a really, really cool paper. But essentially, they took from the tissue blocks, I think, 12 serial sections. They did imaging on some, they did metabolomics on others, and they did the Stomix, and being able to bring all of that information into a single aligned data set through our co-registration, using AI to help define regions, using AI to help identify where all of those cells are, cell segmentation, being able to pull not only biomarker expression from the proteomic side, but also understand what's happening regionally from the metabolomics, where different metabolomes are expressed and high levels, but also now understanding what transcripts at the single cell level are also available in the image. We're also working very closely with one of their partners as they're doing this analysis, being able to bring that transcript information back into the image. So then it becomes available in all of our plotting tools so that you can really understand what's going on at every single cell and be able to visually see it within the images. So it kind of completes our picture of spatial biology, but now I have to add metabolomics in here. And I don't know what the next one's going to be. Maybe lipids. I don't know. It's going to become an interesting space area in spatial biology. So we're very excited about that. And I just want to end on one last note, which is, where do we go from research to clinic, and how do we think about this? At Visiopharm, we're in basic research, we're going broad. We're gathering information from lots and lots and lots of places. And we're going to funnel that down into a targeted panel. And so once that targeted panel is defined, that's when we're going to want to create a standardized algorithm or analytical pipeline so that we can do that reproducibly. And that algorithm is going to get better and better and better so that we can get it to a certain point where we're going to put it into clinical trials, take it into internal and external validations across many, many sites, and ultimately develop an analytical test that can be used clinically, hopefully goes sufficiently through regulatory agencies, but also we'll have companion tests for making sure that the stain of all of those biomarkers is correct and consistent, so that the data coming in is able to be analyzed either by the human, by the computer, or ultimately by both simultaneously with a high level of reproducibility and efficiency. So this is what we do with a lot of our partners as well. So I'm not sure where you guys are in your realm of biomarker research. If you're just looking to see what's going on in the diseased states, trying to understand what normal patients look like, trying to understand how that disorder that the disease causes and how we can restore that through pharmaceutical or other interventions. Or if you're interested in bringing that through a full analytical pipeline, the Visiopharm suite of software is really there to help you wherever your research takes you. We've got solutions to help solve that. So I'd like to thank you for the opportunity again to speak, If you have any questions, I'm happy to address them now. For those of you that are online, I know there's a chat. For those of you that are in the room, I'm happy to address any questions. First volunteer.
I have two questions. One is that bias selection of segmentation, and number two is that AI continues to train itself over time. So the result that we get from AI today, a month from now, it may give us a different result of data. So in one of your talks, you said that we need to trust the result.
Yep.
It could be different a month from now or a week from now.
Thank you. Great questions. Let me see if I can rephrase them so I can remember them. The first question was, how do we avoid selection bias with AI? And the answer is AI, especially if it's trained by annotations by us, it's going to be also as biased as we are in the training and the data set that we give it. Okay, so one way to avoid that is obviously to have training sets that's coming from multiple people, multiple instruments, multiple populations, multiple sites. Now we're building a giant data set that might be harder and harder to accumulate in order to get an AI to solve a simple problem or a small problem because you only have the 15 slides that you want to analyze. And that's why we've created these template apps, because they're built for a general purpose that you can then make a little bit more specific to find your needs. There's other ways of being able to generate AI annotations, however. Some of those are using different chemicals. So, for example, when we trained our AI to know the difference between tumor and non-tumor areas in an H&E, we actually used a stained image that we co-registered. So it was based on a known, our gold standard in that case was stained tissue. And then we can transfer those annotations over using this co-registration technology so that the AI was learning from something that wasn't stained, but from annotations that came from true staining. So now you have to trust your stain, right? The second question was, how do you trust your results if the AI is constantly learning? And actually, our AI doesn't constantly learn. It's you've trained it, you validated it, it stops learning. It only learned from the rules that it was trained on. If you want it to improve, then you have to then apply more data and then tell it to continue to train from a curated data set that you're, again, providing it. So actually the AI that we're using doesn't continue to learn. And we need that in the clinic. We need to lock them down so that they perform at the validation steps that they were at the stage they were validated against. So it doesn't actually continue to learn unless you forcibly tell it to. We have a button that says retrain, so it's actually quite easy. What's really nice about the system that we've built is it doesn't require the old training set in order to continue to train. It can learn from new data, but already remember what it's learned from the previous data. So you don't have to keep that volume of training data available at all times. It's also really nice if you go from one indication to another indication. So it's pre-learned on, let's say, breast cancer, but now you want to use it on lung cancer, which is very, very different. You could have it start from what it's learned from breast cancer and then apply information on lung cancer and say, this is what lung cancer looks like. Use that foundational training that you had in breast cancer, but now learn what lung cancer is. And so there's lots of tricks and things that we can do to overcome some of those challenges, but we don't want it to continue to learn unless we tell it to. We still control the AI. Human intelligence over artificial intelligence. Are there any other questions in the room?
What about the data sets that you upload to the Visiopharm? Take it right off of the vector Polaris or the PhenoCycler? What's there?
Yeah, we work with the manufacturers to read their original data formats, their original metadata. It's all uncompressed, unchanged. We don't want to mess with the original data at all. We'll just read from their native file formats. So it's perfectly seamless with the new Polaris They've removed the need for going through Inform for doing the unmixing. We read those file formats directly. The Fusion, we read their QP-TIFFs directly. We're one of the only companies that read the standard BioTools format right off the bat. We've also incorporated some things where you can do some stitching, because the stitching algorithms are nice to have if you're not doing whole slide images in some of those devices. But we want to read everything from the native file formats unchanged.
When you were talking on geomics, we were talking about the proteomics side of it, or were you talking about transcriptomics?
So we will do the proteomics analysis. We will do the the image-based segmentation. You're still going to do the proteomics the transcriptomics analysis through their system, through their analysis, and then eventually we want to link those together so there's a really good handshake between the two. But our wheelhouse is not understanding the transcriptomics side of things. It's really on the image, and pretty much from the image you're going to get proteomics. The one thing we can do is if you're using something like RNA-scope in geomics, which was never really available because they had no way of thresholding or finding cells that were RNA-scope positive, we would be able to give you that capability now with the different techniques for segmentation there. I don't know if that's something you guys are interested in doing, but it seems to be a growing demand. Okay. Yep. Yeah, the last paper I showed from Sammy and Jared was doing exactly that same thing, using the co-registration to create essentially a single image, multi-layer single image with all of that information. Yeah, I have to do some more homework and learn what some of those techniques are these days, because it's pretty crazy. We do have a question on the chat, so I'm going to read that. It says, I'm curious about oh, sorry, there's a couple. Oh, I did a bad job repeating the questions for the online viewers. I apologize. Would you provide some insight into how you measure the quality of co-registration? Are there any metrics that you report? Ooh, I shouldn't have read that question out loud. Because we are not expecting the two pieces of tissue to be the same, it's really hard to understand what those types of overlap metrics are. If you have, if you're expecting a nuclear to nuclear co-registration, then we can run some dice scores and some other types of scoring metrics to understand the relationship between those if necessary. But really, it's a visual interpretation of what's happening there. And when you're dealing with different pieces of tissue, even the same tissue can fold, can hydrate differently across different staining rounds. When it comes to some of these cyclic staining devices like the PhenoCycler Fusion, we will just read the file from them. So they're going to do that alignment for us directly. I'm curious about the Visiopharm nanostring collaboration. Can you comment on how the AOIs are selected that limits biases in tissue selection? I'm not sure I fully understand that question, but what I can say is we are in the very early phases of this relationship. The integration between the two platforms is done. We are currently beta testing at a couple of sites that use both of our platforms. And I will recommend stay tuned because there's going to be some pretty big announcements at AACR in the beginning of April. I hope that answered the question. Well, if anybody would like to reach us offline, please feel free. We're going to be around this area for the next couple of hours. If anybody wants to come and ask some questions or take a look at some things, we're certainly here to help and talk. But hopefully this is the first of many, many conversations with the single cell spatial group here at University of Michigan. Thank you.
Thanks very much.
And for those of you who are online, we have nothing but cake. Sorry.